Thank you, everyone, for coming, and thank you to Julia and the rest of the Simons Foundation staff for being such wonderful hosts and welcoming us into this beautiful, brand new auditorium. I also want to thank everyone for coming and to welcome the people that are going to be joining us online by seeing the video. Okay, so tonight's presentation is going to feature a short talk from myself and then Ben Philpott, who's going to speak in more detail about one of the approaches that we are using to hopefully cure Rett syndrome. I did a presentation like this a few years ago and hope to update you tonight on some of the developments. A few years ago I didn't need glasses, and now time is marching on and I do. So I'm going to jump right in and explain a little bit about the various mutations that happen in Rett syndrome. And the reason for this is that later on in the talk, I'm going to talk about a couple of approaches where the treatments will be mutation specific. And so I just wanted to run down in a little bit of detail some of the mutations. So the first one is a missense mutation. Proteins are made up of amino acids that are made up of nucleotide bases, and a missense mutation is when you have a mistake in one of the nucleotide bases. And so instead of a particular amino acid, you're left with another type of amino acid. And that small mutation in just one single nucleotide base leads to Rett syndrome. Then you have a frame shift mutation, which basically throws off the sequence of amino acids by either adding a nucleotide base or a couple of nucleotide bases or deleting some. And that's called a frame shift mutation. A nonsense mutation is those of you that have children with mutations that end in X. And that means at the end of every protein, you have a stop codon that basically says to the protein, you're done making protein. And if you have a mutation where the substitution of the amino acid results in a stop codon prematurely, it shouldn't be there, you end up with a nonsense mutation, which is basically in a truncated mutation. And then there are large deletions, which are basically large chunks of the protein that are missing. And I'll refer back to some of these later in the presentation. So one of the questions that I'm often asked, especially by parents of newly diagnosed children, is, now that I know my daughter's mutation, what can you tell me about her prognosis? Will she walk? Will she talk? Will she have some hand function? And it's actually not that easy to correlate mutation with severity. There are other things that influence how that child is going to present. And two main things are the pattern of X chromosome inactivation and modifier genes. Everyone here, I'm sure, knows that girls with Rett syndrome have two X chromosomes. They have the mutation on one, but on the other chromosome they have a healthy MECP2. One they get from their mother, one they get from their father. And all females inactivate one X chromosome very, very early on in development. You know, we're talking at the 5 or 6 day stage. And it's a random process. So in some cells you will have one chromosome that's on, and in the other neighboring cell it'll be the opposite chromosome that's on. Sometimes there is skewing where one X chromosome is favored over the other, and that skewing can either be favorable, where you have more of the normal X chromosome that's on and the mutant is inactivated. In that case, that will lead to a milder form of Rett syndrome, or if the opposite happens and more of the mutated chromosome and gene is activated, you will end up with the more severe version. And a great example of that is a identical twin sisters that I know from the Baltimore area that have the same genetic background. They have the same mutation. One of them has random skewing, so like 50-50, and she has classic Rett syndrome, but her twin has favorable skewed inactivation, and she's much more high functioning. She can talk, she rides a bike, she plays the violin. And so that's an example of what skewing can do. The other phenomenon that can happen is modifier genes. Every disease has modifier genes. So for example, if you think of two people who smoke 2 packs of cigarettes a day, one of them gets lung cancer, the other one doesn't. The one that doesn't has, in their own genetic makeup, probably mutations that are advantageous and protect them from getting lung cancer. You see the same thing with cholesterol. People can eat very high fat diets and have low cholesterol. Their genetic background gives them that ability. So these 2 pictures were taken from a publication that just came out in a journal called PLOS One from a geneticist in Italy. And they are sisters with each picture shows 2 sets of sisters. They have the same mutation. They have balanced X inactivation, so it's not skewed, and yet they present quite differently. One of the sisters in each of the pictures has classic Rett syndrome, and the other sister is much more high functioning. And so we think that it is modifier genes that are behind that. So modifier genes is an important aspect in Rett syndrome, and I'm going to come back to that. So this is an important slide. This shows all of the approaches that RSRT is currently pursuing to treat and hopefully cure Rett syndrome. And we're going to talk in some length about each of those. But for now, take a look at the 3 main buckets: MECP2 as the target, suppressor genes, which is the modifier gene approach, and then downstream targets. And then I'm going to talk to you about each of those areas. And then off to the far side is basic research, meaning we are still trying to figure out what MECP2 does, and we are funding projects including the MECP2 Consortium, which are 3 labs working cooperatively trying to figure out what this protein does, in the hopes that an understanding of this protein will open new doors into how potentially to treat it. So the first thing we're going to talk about is activating the silent MECP2. I'm not going to go into a huge amount of detail because that's what Dr. Philpott's going to be speaking about. But I do want to tell you why it's an approach that we're particularly interested in. So I like this approach because it really attacks Rett syndrome at its very root and not downstream. The problem in Rett syndrome is an MECP2 mutation. And there is a healthy copy of MECP2 in every cell of a girl with Rett syndrome. About half of those cells are shut down. If we could find a way to turn that cell, that gene, back on, in essence, we treat and hopefully cure the disorder based on the Bird reversal experiments. We don't need to deliver a gene as we would need to do in gene therapy. It's already there. We need to turn it on. Also, there is a concern that in Rett syndrome, too much MECP2 is a bad thing, and we know this from the MECP2 duplication syndrome. These are kids that don't have a mutation in MECP2, but they have an extra copy of the gene, and that leads to a neurological disorder that is quite severe. And ironically, it's found mostly in boys and not girls. But with this approach of activating the silent MECP2, we don't need to worry about the dosage. And finally, we don't need to know the function of MECP2 to turn it back on. After quite a number of years of research, the function of this protein remains rather elusive. And with this approach, we don't need to know that. We don't need to know what the downstream targets, downstream genes are. So I have here 4 of the labs that we are funding that are attempting this approach. Each of them is using a different kind of assay, and they're using different molecular libraries, different compounds that they're testing. And Ben's going to talk in more detail. I will say just a couple of sentences about Michael Green's project. This is a new project that we committed to fund this week, actually. It's a $750,000 project over 3 years. And he's taking a different approach. Rather than turning on the silent MECP2, he's actually looking to turn on the entire X chromosome. And this is a bit radical and controversial, actually, in the X inactivation field, because dogma tells you that we don't really want to do that. It might be a very bad thing. He has data to suggest that maybe it might not be as bad as we thought, and perhaps activating the X chromosome later in development is not detrimental, whereas activating it during development or early childhood might be. So we don't know. It's the kind of experiment that the kind of project that you don't know until we try it, so we were willing to invest some funds and see whether activating the whole chromosome might be beneficial. So that's a new project. Okay, now we're going to move to gene therapy. So gene therapy's had a bit of a rocky road. The first gene therapy clinical trial was in 1990, and so far in the US, there are no FDA-approved gene therapy treatments yet. There was a treatment that was just approved in Europe, and there's a treatment that's been approved in China for a few years. But the last few years has seen dramatic improvements in the field and dramatic progress. So, you know, I think there's reason to be optimistic. So there are a number of labs that you can see on that chart that are working on gene therapy approaches in Rett syndrome. And the concept is pretty easy. You have a mutation in a gene, and with gene therapy, you try to deliver a healthy copy of the gene into the cells. And the way you do that is with something called a vector, which acts as a Trojan horse. There's different kinds of vectors, each with their own profiles in terms of how you deliver them and what type of cells they infect. We had a meeting in Westchester County in November where, for the first time, all the gene therapists that are working on Rett syndrome got together for a couple of days and shared data. Different labs are getting different results. I don't think a clinical trial for gene therapy is around the corner quite yet. I think there's more basic science. But I think there are some preliminary data that suggests that this might be a feasible approach. The next slide, I'm going to talk about 3 approaches. The first one is boosting wild type, which means normal MECP2 levels. So, as I mentioned before, girls with Rett syndrome are mosaic for MECP2. That means that they have normal amounts of MECP2 in some cells, and they have mutated, which probably means low levels of MECP2 in most cases, in the other cells. So Huda Zogby, who was the investigator that identified MECP2 mutations as the leading cause of Rett syndrome, came to us with the project and with the hypothesis, wanting to test the hypothesis, what would happen if we could boost the levels of MECP2 in the cells that already have the normal amount? Could that somehow restore the network connections? We don't touch the cells that are mutated, but we increase the level of MECP2 in the cells that are normal. So those experiments are ongoing. If she finds that, in fact, it's not toxic and beneficial, then we would move to the second step of the project, which would be to do a screen to look for compounds that can boost those levels for Rett syndrome, and at the same time, look for compounds that would decrease MECP2 for the duplication syndrome. Read-through compounds. Read-through compounds are a class of drugs specific for the nonsense mutations, which is why I wanted to describe the nonsense mutations in the first slide. So these are for the mutations that end in X that have the premature stop codon. And these are compounds that can basically eliminate that stop codon. And they actually substitute a different amino acid. Chances are it's not going to substitute the amino acid that should be there, but it'll substitute another one, and hopefully the end result is a less severe symptoms than you would have with the premature stop codon. Okay, and then we have pharmacological chaperones. This is a class of drugs that targets mutations that lead to misfolding. So when there is sometimes when there is a mutation in a protein, what ends up happening is the protein, which is supposed to be folded in a very specific way, can't. And so you end up with a misfolded protein. And these pharmacological chaperones are designed specific for a particular mutation to help refold that protein the way it should be. So this is still a very, very, very early stages in Rett syndrome, but there was one paper, actually a couple of papers, one by Adrian Bird, showing that the most common mutation in Rett syndrome, the T158M mutation, leads to a misfolding problem. And so pharmacological chaperones could be pursued to help reshape and refold that protein properly. So this would be something that would need to be pursued for individual mutations. First, we would have to test which ones lead to misfolding, and then you have to develop compounds specific for that mutation. As opposed to some of the other approaches, gene therapy, activating the silent MECP2, that are much more generic. They would work for any of the mutations. Does that make sense? So this is a project that RSRT has been funding for a while now. It's being pursued in the lab of Monica Justice at Baylor. She is a mouse geneticist. And this was a project that came out of a brainstorming session at a Rett Syndrome meeting that I organized in 2006. And it was a meeting of about 100 people, and I gathered about 24 people for a very early morning. I think it was like 6:30 in the morning, pre-meeting meeting. And what I asked them for was to kind of come up with high impact, high risk type projects that weren't currently being done that could kind of move the field forward. And they had a few projects, and the one that came out kind of at the top of the totem pole was a modifier screen. So this would be a project that's done in mice to try to identify genes mutations in genes that make having the MECP2 mutation less severe, like what I discussed earlier with the two siblings. So the nuts and bolts of this project, when it was first described to me, sounded like a real needle in a haystack, but actually it's turning out to be very doable and is starting to yield results. So what they do is they give male normal mice, not Rett mice, but male mice, a drug that induces mutations in the sperm. And then they mate these mice with female Rett mice, and then they watch the offspring. A certain percentage of them will have inherited a random mutation from the father, and they will have an MECP2, no MECP2 that they get from the mother. And then you watch these offspring, and you watch for any mice that behave better and have less symptoms than your typical Rett syndrome mice. And after thousands and thousands of mice, right now Monica Justice is about 15% of the way through the screen, and she's identified 5 modifiers. One of which is in a pathway that we know a lot about, that drugs exist. She's already tested these drugs on the mice with improvements. And so it's a real proof of concept that this can work. So we've just committed funding for her to get to the 50% point of the modifier screen, which she should do within 2 years. The cost of sequencing and trying to identify these modifiers is coming down. So at the 50% mark, we'll reevaluate and commit for the next 50%. And what we're hoping is that in a matter of about 3 years, we will have a list of all of the modifiers, at least in mice, that exist. At that point, we can prioritize that list, look for genes that are in pathways that we know something about and that drugs exist. And that could potentially lead to clinical trials if there are FDA-approved drugs already rather quickly. What I like about this approach is it's very unbiased, and it will also lead to a better understanding of what MECP2 does and the various pathways that it's involved in. And so far, some of these modifiers that she's identified would never be modifiers that we would normally think that could interact with MECP2. The next one is basically everything else. So downstream targets. So these are drugs that we think could improve some of the symptoms. Probably these are not going to lead to a cure for Rett syndrome, but they may improve some of the symptoms, the breathing, the seizures, the movement. So for example, IGF-1 would fall under this category. The Neuron drug, the trial that's being started at Baylor, would fall under this category. All of the many papers that you hear about, the ketamine, the drug from David Katz's paper that came out recently showing that an anesthetic might be helpful. So that would fall under this category. And so basically what this involves is testing in the animal model, or sometimes if the mechanism is well known and if the drug is already FDA approved with a very safe profile, it could potentially go right into the children in a small clinical trial. I did want to just spend just a brief minute or two talking about some of the challenges when doing mouse studies. And some things for you to keep in mind when you're looking at, when you're reading papers or reading press releases of papers. So one of the things that I think is important to notice is whether the study that was done talks about a reversal of symptoms, meaning symptomatic mice that were given a drug or a procedure that made their symptoms go away, versus something that stops symptoms from happening. And so, for example, if you're reading a paper or looking at a press release and they're talking about 2 or 3 week old mice, those are pre-symptomatic mice. So they could have stopped the symptoms from happening or slowed that process down, which is different than taking mice that already have the symptoms and trying to reverse those symptoms. Also, if you are reading the Rett literature, you'll notice that most of the experiments are done on male mice and not female mice, and there's a reason for that. The reason is the male mice develop symptoms earlier, around 4 to 6 weeks. The female mice don't develop symptoms until 6 months. And the male mice are much more severe and they die, whereas the female mice live a normal lifespan and their symptoms are milder. And so from a cost perspective, but also from a ease of detecting improvements, it's easier to do experiments on mice. And so most scientists will actually only stick to mice. Some of them will test whether something works in a mouse, and if it does, they'll do it in females. But I'm often asked that question, so I thought it would be something to bring up. The other thing that has been getting a lot more attention, I think, in the scientific literature in general, not Rett specific, but just in general, is the importance of doing really rigorous studies. So that means with statistically powered, meaning enough mice so that you can detect differences, and that scientists are doing these experiments blinded and randomized, and that a lot of care is taken, and that also work needs to be independently confirmed before, to kind of validate it. And so these are things to kind of, just to be aware of. I wanted to just put up there a slide that kind of shows what the drug development timeline looks like. If you're starting with, creating a drug from scratch, you've got typically 5 to 10 years of basic research, and then it moves to preclinical testing, which means in mice. Anytime you see preclinical, that means mouse work or some kind of animal work. And then a phase 1, which is usually just a safety test. You're not looking for efficacy. You're not looking to see if the compound is actually helping, but you're just making sure that it's not toxic. And then you move to a phase 2, which is a small number of patients, and there you are looking for efficacy and also safety. And then finally a larger phase 3 where you're looking for efficacy in larger groups of people. And we don't necessarily have to start from scratch for every treatment approach that we're using. So for example, if Ben Philpott's project ends up yielding some compounds that can activate MECP2, if it's an FDA-approved drug, we're not starting from scratch. We might be able to jump in at a phase 2. If there is a drug that, like the Neuron drug or IGF-1 or, other things that are being, ketamine, for example, we can jump in at a phase 2. We're not necessarily starting from scratch. Now, if we're developing a drug for a specific mutation, that's going to take longer. So when people ask me, how long is it going to take for a cure for Rett syndrome? It depends, and I wish I had the answer for that. I don't. It kind of depends, and it's not one answer. One size does not fit all. These are the current clinical trials that are ongoing right now. The industry program, I have these divided as academic programs and industry programs. There's a trial that's starting in Italy with a drug made from a company called Edison Pharmaceuticals. This EPI 743 is a drug that's used for mitochondrial disorders and is going to hopefully treat some of the oxidative stress that we see in Rett syndrome. So that's starting now. In Italy, they're recruiting patients. And then there's the Neuron drug that is starting at Baylor, which is very similar to IGF-1. It is an oral drug instead of an injectable, and it is supposed to cross the blood-brain barrier better than IGF-1. So that's going to be an open label study for 10 kids, I think, and they're adults actually with Rett syndrome, and they're recruiting now. And then we have a couple of academic programs. One of them is the IGF-1 trial that's going on in Boston. The other one is the dextromethorphan trial at Kennedy Krieger, Johns Hopkins. And then one that RSRT just committed funding to, and that's going to be recruiting patients. This is a drug called Copaxone. That is a drug that's used in MS. And the thought behind this is that it increases BDNF. BDNF is a growth factor that we know is deficient in the Rett syndrome mice. And there's numerous studies showing that if we can boost BDNF levels, it should help the symptoms. And so this will be a 6-month trial with 10 kids to start with to test whether it helps. If it does help, then we'll move on to a larger trial.
Is this being done by Sasha?
Yes, this is being done at Montefiore. We are also going to be funding one in Israel for also a small number of kids. That way we can kind of validate and confirm things independently in 2 labs right off the bat. We're still waiting for some IRB approvals, but that should be coming shortly. So what I wanted to end with is that we are actually entering a golden era for rare diseases and pharmaceutical companies. I mean, typically we think of, well, Rett syndrome is rare, there's not a lot of kids, so pharmaceutical companies won't be able to make a lot of money because the population is small. You know, how are we ever going to get a drug for Rett syndrome? And actually, pharmaceutical companies, which until recently have been very devoted and interested in blockbuster drugs and things like Viagra and Prozac and statins, are now looking and becoming much more interested in rare disease. And every large pharmaceutical company has a rare disease division now. And there's various reasons for this. One of them is they are encountering a pipeline problem, meaning a lot of the blockbuster drugs are hitting the end of their patents. They're coming off patents, which means drug companies are going to lose a large source of revenues. And they're looking for other things to make up for this revenue stream. And there's high unmet needs in rare diseases. There's typically not a lot of competition. And there's a lot of perks as well. Orphan drug status gives you longer patent protection. Doing clinical trials for a rare disease is much, much cheaper than having to do a clinical trial, say, for a statin. And so it costs a lot less money. Might also be easier to recruit patients because patients are more eager to get enrolled in clinical trials. There's faster FDA approvals. And the price issue can be figured out because with a smaller population, you just charge larger prices. And so rare diseases is actually an area that's very, very hot right now in industry. And I think that that's a great opportunity for us. I've had more conversations in the last 6 to 9 months with industry than I've had in 14 years put together. And Rett syndrome has some really key advantages. We're a 1-gene disorder. We have really good animal models. There's no neurodegeneration. You can have reversal, at least in mice, even in late-stage disease. So these are a lot of advantages that industry is looking for. And so they're not quite ready to put their own money into Rett syndrome programs right now. But hopefully that will change. And I see the role of organizations like the Rett Syndrome Research Trust is to cultivate these programs and bring them along until pharmaceutical companies are willing to partner with us. And basically we have to de-risk. They don't want to take a lot of risk early on, but if we can invest money and move the science along until they feel more comfortable, then they partner and then they can put in a lot more money, but they have to feel safer, that they're not taking too much huge risks. And that's really what our role is. Any questions?
Just one other question in the past. You said, would modifier genes work on all types of mutations, or would it work on one type of mutation over another?
That's a good question, and I guess specifically we don't really know, but I would. It should work more broadly. It shouldn't be specific, but I guess until we really uncover these and then figure out the mechanisms of the interactions between these modifier genes and MECP2, we won't really know.
Then this something's a pie in the sky question, because there's different techniques. What happens if our doctor, there's coming up with different solutions at different times? Could you actually, if you're doing a treatment for modifier genes, could you do, say, another treatment for?
Well, you couldn't be in multiple clinical trials at once, of course. And I think that each clinical trial will have its own set of criteria. They may want you to be, treatment-free for X number of months, or, a year after you've been in a trial. I don't know. It'll depend. I don't think there's a generic answer to that. But you do have to, when you go into a clinical trial, you do want to make sure that any benefit that you're seeing is actually coming from that treatment that's being tested and not for some other reason.
Do you think that scientists or doctors are going to be looking at combining treatment?
Yes, I think absolutely there could be a combination. So I could envision a situation where we find a drug that can activate the silent MECP2 in a certain number of cells. We may not hit every cell, right? And in Rett syndrome, you have to hit a fair number of cells. But if we could combine that with perhaps gene therapy or a procedure, deep brain stimulation, a bone marrow transplant, something else, it could be a combination approach. Ideally, it won't be. Ideally, we'll have one method that works, but it may not turn out to be that way. And especially when you're looking at the downstream genes, where you're looking at various drugs that do different things, it may be a combination approach.
I know she didn't have the bone marrow.
So the bone marrow transplant, as most of you know, we've been funding confirmation studies. So these are other laboratories that are doing the same experiment, trying to validate, and those aren't done yet. So there's not much I can say about it until they're done. We're also still waiting for the female reversal data. So is this a situation where you have to intervene early and stop symptoms from happening, or can you intervene late and improve symptoms? So those are still ongoing.
Would that be considered gene therapy? No.
No.
Not. What would you put it under?
Well, it would be under downstream. Yes, it's not a drug, it's a procedure. The other thing that would fall under procedures is deep brain stimulation. So there's a couple of labs so deep brain stimulation is a procedure that's being looked at for Parkinson's and depression. And basically they put electrodes into the brain and provide some stimulation. That's something that's also being tested in the Rett mice. So it may be a combination approach. Okay, finally I just wanted to thank you all for coming, thank the Simons Foundation again, and also to thank people that work really hard to raise money that makes this work possible. So the various organizations that partner with us, the Rett Syndrome Research and Treatment Foundation in Israel, RSRT UK, Girl Power to Cure, the Kate Foundation, our trustees, people that raise money for us, many of you who are here in the room today. We couldn't do any of this without you, and we thank you.